Hi everyone, thank you for coming to my poster presentation. Before we get into the awesome science stuff, I just wanted to display this image and thank our sponsors, because without our sponsors, AEC would not be possible. And also I would like to shout out the Roy Lab because Megan Hoffman is an alumni of the Roy Lab. All right. The title of my poster is Transgenerational PUE of Daphnia in Eutrophic Media. I'm sure some of you who are familiar with the Roy Lab might have a general idea of what I've been doing, but I would like to explain a few things before I dive right into it. First, I would like to explain what eutrophication is. Eutrophication is an excessive buildup of nutrients such as phosphorus and nitrogen in freshwater systems as a result of agricultural runoff. The result of this is often a massive chain of issues, starting with the development of algal bloom. You can actually see an example of algal bloom in figure 2 right here. The green layer on top of the water, that is eutrophication and algal bloom. And this is often toxic and can cause hypoxia underneath the bloom, which kills several aquatic organisms. In fact, recent evidence suggests that about 95% of lakes in North America are eutrophied. So I wanted to test the impact of eutrophication on lake organisms over an extended period of time. In this study, Daphnia is my model organism. This is an example of a developing Daphnia. They are keystone grazers in freshwater systems. They consume algae, and they are also food to larger fish. Daphnia are sensitive to chemical changes in their environment, so they are excellent indicators of the health of a lake or a pond. If they are not present in a body of still water, it's indicative of a deeper issue. They have a very high body P content, making them particularly vulnerable to eutrophication. For my study, I set out to test the changes in somatic phosphorus or P content and P tolerances in Daphnia that are exposed to 10 generations of simulated eutrophic conditions. P tolerance is measured as phosphorus use efficiency, or PUE, the amount of biomass produced per unit of body P concentration under P limiting conditions. In other words, if I expose my Daphnia to eutrophic conditions for 10 generations, will they become careless with how they process and use phosphorus? I hypothesized that the transgenerational exposure to simulated eutrophic conditions will increase the Daphnia's somatic P content and decrease PUE due to the excessive environmental supply. To grow my generations, I created a media that simulated high P or eutrophic conditions. The media was 1 part high P media to 9 parts no N and P combo media. Upon testing, this media was shown to have about 150 micrograms of phosphorus per liter. Eutrophic conditions are generally expected to be at or above 100 micrograms of phosphorus per liter. And we grew our cultures in glass jars at 28 degrees Celsius with a cycle of 16 hours of light to 8 hours of dark. Once 10 generations were obtained, I tested PUE and P content. We measured P content using a sulfuric acid molybdate process first published in 1998 for zooplanktons. This process takes 3 days and involves drying the Daphnia overnight, weighing them on a microbalance that gives precise measurements, placing them in a furnace at 550 degrees Celsius for 2 hours to combust the organic parts, and then adding sulfuric acid, ascorbic acid, and the molybdate reagent that gives a blue color upon reacting with the phosphorus in the sample. We measure the intensity of this color using a spectrophotometer. Following Beer-Lambert's law, the intensity of the color is directly proportional to the amount of phosphorus present. We then use a standard curve, as you see in this picture, obtained from running known quantities of NIST spinach, standard, for P to estimate the P content in our unknown media. And this is a condensed methods section. So if you would like to know exactly step by step the protocol that I use, I encourage you to check my references. So to analyze our data we obtained from these experiments, we performed an unpaired t-test right here for somatic P content and PUE. As you can see in figure 6, our results show that there is a statistically significant increase in the somatic P content from generation 0 to generation 10. If you look here, you will see the graph for the phosphorus use efficiency of F0 and F10. While our p-value was not statistically significant, it is clear that there is a decrease in PUE in F10 compared to F0, indicating that Daphnia do become careless when a lot of phosphorus is available in the surrounding water. We see a lot of variation in the PUE of our F0 generation, which is indicated by the larger standard error bars right there. So we are testing additional replicates to further validate these results. My results do indicate a shift in somatic P content and probably a decrease in PUE in a keystone grazer when exposed to simulated eutrophic waters. This is significant because body phosphorus content is usually a homeostatic trait that organisms tend to remain stable in, and any changes in these can impact phosphorus cycling in freshwater systems via consumer-driven nutrient recycling. In the future, I would like to expand the experiment to include genotypes that differ naturally in their body P content, test natural populations that are exposed to different P regimes, and also understand the interaction between temperature stress and increased P supply in these keystone grazers. Thank you for listening to my presentation. I would also like to thank AEC for allowing me to present my data in my experiment. If you have any questions, please don't hesitate to reach out and email me at autumn.berlied@ksc.keen.edu.